The acquired immune system is split up into cell-mediated immunity, which directly attacks infected cells, and that is the responsibility of T cells or T lymphocytes. And the other component of acquired immunity is known as humoral immunity, and that's based on antibodies which are secreted into the bloodstream. Historically or early on in the studies of human biology, blood was referred to as humor, hence humoral immunity. And humoral immunity is the workhorse of antibodies, and that's what we're going to talk about in this video. Immunoglobulins and antibodies are the exact same thing. There's a number of different types of antibodies. There's IgG, IgE, IgA, and a number of other antibodies. But for this video, we're just going to be talking about antibodies in general. Okay, so here we have a B cell, and B cells, like all lymphocytes, need to become immunocompetent. That is to say, they need to learn how to recognize foreign antigens. And upon recognition of foreign antigens, they produce these cell surface protein receptors, otherwise known as antibodies. So these are antibodies on the surface of a B cell, and they are specific to a certain antigen. And let's just say here's the antigens right here, which would be on the surface of a foreign cell or foreign organism or a virus. And these antigens would bind to these antibodies. Those antigens bind to those antibodies because the antibodies are very specific to them. They have been designed or structured to allow that antigen to bind to it. So B cells, just like T cells, we talked about the division of lymphocytes into memory cells and effector cells. When B cells divide, they are going to produce memory cells, which will remain in the body to provide a quick response on subsequent exposure to an infection. So historically, before the chickenpox vaccine, otherwise known as varicella, children would get the chickenpox and generally not get it again because they've developed memory cells to the varicella antigens. Now, back in the day before the vaccine, it took roughly 2 weeks for the development of these B cells to produce these antibodies specific to the varicella antigen before the acquired immune system can actually attack and rid the body of that varicella virus. So there are memory cells that will stay within the body for subsequent exposure. But when a B cell divides, it also produces effector cells, which actually carry out the work of identifying foreign antigens. And that's really what antibodies do, is that they highlight foreign antigens to make them more visible. They highlight pathogens to make them more visible for the subsequent work of T cells. So the B cells are going to secrete antibodies into the bloodstream. They will flow through the bloodstream, and whenever they come in contact with their specific antigen, that antigen will bind to the antibodies, and then the antibodies, the antigen-antibody complex will then be highlighted for destruction by T cells. And that's the very basic premise of antibodies. Once again, B cells initially produce the antibodies, they become immunocompetent, they will divide into memory cells, which are the more B cells, and the plasma cells, which are nothing more than the effector cells. And the plasma cells will secrete antibodies